Okay, so we're going to pick back up where we were. And again, just in case we couldn't hear some of that from the last video, this region where there were only thin filaments is the light band or I band. This area right here where we had only thick filaments was the dark band, but specifically the H zone of that dark band. This also is the dark band or A band. Those words mean the exact same thing. Specifically, this is in the H zone, but within the H zone, it is in the M line. You can see these proteins holding the filaments together, so we know it's within the M line. And then this one again had both thick and thin filaments in it. And so because it had both, we know that we are looking at the dark band because we have thick filaments. So anywhere there are thick filaments have to be in the dark band. But because it also has thin filaments, we know it is in the outer edges of the dark band. And what I mean by that is it's anywhere other than H zone. So anywhere down this plane, anywhere here, or on this side that would be in the dark band. It just cannot be in this place, cannot be H zone. Okay, so let's move forward a little bit more. So what we're seeing here is a portion of a thick filament. So if you look at it, let's go back over here. So we're looking at basically a portion right here. Okay? A portion of a thick filament. And what thick filaments are made up of are myosin. And so here are two myosin molecules that are bonded together. And myosin is a contractile protein. And it is a filamentous or fibrous protein, which means that it is long in nature instead of globular. And so a couple things that I want you to realize is that these have some binding sites on them. Okay, these little holes here, these little darker areas are considered binding site. And what that means is a binding site is an area where another molecule will attach. And on the heads of the myosin, that's what these are called. So you have two myosin heads and two myosin tails that coil together to form this myosin molecule. These binding sites are actin binding sites. So the thing that will bond in that space is actin. And then the other thing you have over here, one here, and there's one on the other side too, you just can't see it. This is an ATP binding site. What happens is about 300 or more of these myosin molecules will arrange themselves together. And they kind of coil together, and they do so where that the heads point out and the tails point towards the middle. So if we're to go back and look at this picture, you can see this is an area where it's just tails. There are only tails there. And the heads point out this way, and heads pointing out that way, head pointing out that way. On this side, same thing, tails here, but heads pointing out that way, tails with the head pointing out. And so that's the way that they arrange themselves. And so the thick filaments truly are just made up of myosin. It's just made up of hundreds of myosin molecules arranged in that area. And be sure that you are aware that these are the myosin heads and that they have binding sites on them. And the binding sites here are for actin, and this one, you can't see the other, but it's on the other side, is for ATP. And also keep in mind that at this point right here, myosin has the ability to pivot. The heads of that can actually move or rotate on the tails. Okay, so what we have here is this is a part of the thin filament. So let's go back and look at the last diagram. So we're looking at this. We're blowing it up. And when we see that, we can see there are a few structures that make this up, a few components. So each of these individual beads, these little blue beads that also have binding sites on them, see these nice little divots, those are the binding sites. These are called G-actin, letter G, and G stands for globular. And the reason it's called globular is because they are shaped kind of like a glob, as opposed to being filamentous like what myosin was. And what happens is these G proteins attach to each other and make long chains like this. So here's a chain, and then here's another chain. Those long chains, each long chain is called F-actin, F meaning filamentous or fibrous. And so the long chains are the F-actin, and each individual bead that makes it up is called G-actin. And to make a thin filament, two long F-actins will just coil around each other or twist around each other. So that's what the blue portion is, and that is the majority of the thin filament. However, there are two very, very important accessory proteins. And here you have troponin. That's the yellow, and you can see multiple of the troponin on this. And this would continue on. It would be much longer than this. And then attached to the troponin, you have this kind of orange fibrous protein that is blocking all of the active sites or all of the binding sites. Those are the same thing, just different ways to say the same thing. What happens is troponin has three different parts to it. One portion is the calcium portion. It's going to have a calcium binding site. One portion is bonded to troponin, and then the third is actually bonded to the actin. And basically, troponin is kind of like tack or glue that holds those three things together. So troponin can be attached to calcium. At the same time, it's also attached to troponin and to the actin, and it kind of holds everything in place. We're going to come back to that soon. Okay, so now we're going to kind of go back a little bit in the structure. So we've been looking at each individual filament, which those are these things over here. We're going back now to where we're looking at a bundle of myofibrils. So this structure over here is a muscle fiber or a muscle cell. And we know that those are made up of many, 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 many myofilaments. So we have pulled a few of those myofilaments out here to where we can see them. Okay, so here they are. We can see there's some mitochondria in here. We know that's important for muscle contraction. This portion out here, this white part, is the sarcolemma that you can see out here that would have been around the cell. And then we have 5 myofilaments here. Hopefully you notice there's some new structures on here we haven't talked about yet. But also, if you look at this, you should be able to identify many of the parts of the sarcomere. Okay, these blue structures are called the sarcoplasmic reticulum. We've written that name down in your notes before, but we haven't talked about it in much detail. It is one of the special organelles that muscle fibers have. And so what it is a modified smooth ER. So if you remember the smooth endoplasmic reticulum that you learned about in your first year bio class, this is one that has just been adapted to have a specific function within muscle. And its entire job is to house calcium. So it holds calcium. And basically it's just a bunch of membranes, like membrane tubes. And inside of it is lots and lots and lots of calcium ions. Now the end of it right here, there's another end over here. Here's another one, and it has an ending to it right there. So we're talking about the part that is horizontal, not the vertical. I'm sorry, that is vertical, not horizontal. This part is called the terminal cisternae, and it just is the end of the sarcoplasmic reticulum, and it lines up right along the Z discs, very close to the Z discs anyway. Okay, so those are the terminal cisternae, and that's going to be important because that is where calcium is going to be released. So now one other major structure you need to see here, and it's right there between those terminal cisternae. You can see one there, and you can see one here. It's the same color as the sarcolemma, and that is for a reason, and that's because it is part of the sarcolemma. In muscle fibers, the cell membrane does not just stay on the outside of the cell. It actually pushes down into the cell. And so this is actually part of the membrane that's coming down into the cell. That is called a T-tubule. It is where the membrane from the outside, so if it was out here, actually pushes into the middle of the muscle. It's a way that we can get signals that are traveling along the sarcolemma to get into the center of the muscle fiber. So the last little terminology you need to know from this screen, and then we will move on to the next, is that when you have one terminal cisternae next to a T-tubule, and on the other side you have another terminal cisternae. There's 3 parts there. So terminal cisternae, T-tubule, terminal cisternae. Those 3 things together are called a triad. That is a triad. Okay, so we're going to stop the video right here, and I'm going to create another one that starts with the muscle contraction. But those are the parts of the muscle, and you will need to be able to identify all of the different parts of the muscle. So spend some time. You can find these pictures online. You have them posted on Google Classroom. You can find lots of different ones. Spend some time being able to label these parts. That will be a big part of your test.